Thank you for having me, and a special thank you to the organizers of the Australian Space Biology and Health Symposium for providing me the opportunity to share with you all my efforts in relation to our human space exploration endeavors. It's a great honor to be here with you all today. For today, I will share how our studies of our cells adapting to space provide us a unique perspective not found anywhere on this planet. As we reach for the stars, not just to learn more about our universe, but through this journey, we also learn more about ourselves. As we now aim to return to the moon with our international collaborative efforts through our Artemis project, not just to visit, but to stay, there are many questions of how we adapt that remain unanswered in the context of astronaut health. To begin, a mission to the moon will feature new factors compared with previous spaceflight missions. Spaceflight studies to date have explored low Earth orbit, or LEO, exposure from the Space Shuttle era and the International Space Station. The predominant environmental factor in LEO compared to Earth is a lack of gravity, or microgravity. In LEO, there is also space radiation. However, there is protection from space radiation while in LEO through shielding from Van Allen belts. Contrastingly, a lunar mission will have deep space radiation effects, as the moon is located beyond the Van Allen belts. The lunar environment also will have lunar gravity effects, and currently we have minimal knowledge of either factors on the cardiovascular system. So what is the cardiovascular system? As illustrated in this diagram, the cardiovascular system includes the heart, arteries, veins, and lymphatics. The heart, arteries, and veins transport and serve blood to and from all organs of the body, with blood containing macro and micronutrients, oxygen, water, and carbon dioxide. The lymphatic system transports lymph, which contains excess fluid not collected by the veins, and also importantly, components of the immune system. Lymphatics transport lymph to local lymph nodes, which utilize the immunological information contained in lymph to develop immune responses. The focus of my presentation will be on the arteries, veins, and lymphatics. Moreover, there are multiple adaptations being in space. For one, as illustrated here, on Earth, our bodies have a hydrostatic pressure gradient of 200 millimeters of mercury at the feet and 70 millimeters of mercury at the head. Now, during microgravity exposure, astronauts experience a cephalic fluid shift, causing this gradient to become 100 millimeters of mercury across the body. This fluid shift causes significant adaptations of the vascular system in terms of structure, regulation, and function. Each branch of the vascular system adapts differently, of which our knowledge, even with regards to low Earth orbit adaptations, is limited. Now, as the illustration describes, there are systemic organ adaptations to the spaceflight environment, from weight-bearing musculoskeletal decrements, to alterations in kidney filtration rate, to sensory motor adaptations, and immune dysfunction. All organs rely on the vascular system to support their functions, so in turn, adaptations of the vascular system will affect all organs and their functions. Additionally, do all organs adapt independently in spaceflight? There is increasing evidence, from my studies as well as from many others, to suggest not entirely. For example, there is evidence to suggest an integration between adaptations of the cardiovascular system, the gastrointestinal or GI system, and distant organs from the GI, such as bone, as illustrated here. To explain this further, the GI system serves as a barrier and absorption organ for ingested food and liquids, selectively absorbing macro and micronutrients by local GI, venous, and lymphatic capillary beds that would ultimately reach systemic circulation and in turn all organs of the body. Selective absorption also provides time for the GI's local immune system to sense, respond, or adapt to foreign contents. If the GI immune system is overly stimulated or suppressed, as some evidence of spaceflight exposure suggests, GI function will be impaired, which will impact all organs of the body, as all organs are reliant on the absorption and transport of nutrients and fluids by the GI. Furthermore, the GI immune system is constantly producing immune factors that reflect the GI state. And if the GI is in a state of dysfunction, inflammatory immune byproducts are produced, and in turn, transported systemically to distal organs, such as bone, that may in turn respond to these GI immune byproducts. Now, why is this important? Well, the function of our digestive system affects our entire body. The digestive system not only breaks down what we eat and drink, but importantly absorbs everything we ingest, to then provide these materials to sustain every organ, health, and well-being. So in turn, if the digestive system is negatively affected, and its absorption function negatively impaired, then every organ in the body is negatively impacted. I also wish to mention that the physiology is related to nutrition, as dietary composition of what we eat and drink relies on the digestive system to absorb the content of our food. And again, if absorption function is impaired, what does a healthy diet matter if the diet cannot be absorbed and have its effect? So to answer this line of inquiry, I and others have ongoing studies to gather knowledge of these topics in the context of simulating an Artemis and Lunar Mars mission. From these studies, we will discover for the first time and learn how the digestive organs and many more adapt as we prepare for a return to the moon. Now allow me to switch gears and describe some of our current knowledge of GI, cardiovascular, and immune state findings in response to spaceflight environmental factors. All data highlighted in gold are for control datasets, with those in garnet for simulated microgravity. In one study utilizing ground-based analog model for simulated microgravity, the rat hindlimb tail suspension model, GI structural damage was observed as described by this graph. Briefly, the y-axis is the level of GI structural damage, 0 being no damage, 16 being the max level of damage quantifiable. The simulated microgravity group experienced elevated levels of GI damage compared with controls. This was further studied by measuring lymphatic and immune markers by immunofluorescence labeling of GI tissue, with representative images here. The lymphatic marker is in green, and the pro-inflammatory immune cytokine is in red. In quantifying these markers, the level of pro-inflammation was increased in the simulated microgravity group, with the level of lymphatics present in the tissue also elevated, showing that microgravity has an effect on both lymphatic and immune factors. Now, are there also adaptations of the vessels that serve and support the GI? I and others can test the physiological function of the arteries, veins, and lymphatics by dissecting said vessels from these tissues and conducting what are called myography experiments. The images on top left here illustrate the location of the vasculature associated with the GI system, and from the same study I previously was mentioning, I saw an effect of microgravity as described in the figure in the middle. This graph is a mathematical description of the volume of lymph transported by mesenteric lymphatics at physiological pressures, with an elevation of ejection fraction seen at all pressures in response to microgravity exposure. Other studies exploring low Earth orbit spaceflight effects include experiments from the space shuttle of arteries, veins, arteries and veins, which are represented by these two figures on the right. These show impaired vasoconstricture regulation of both vessel types immediately post-spaceflight. In conclusion, there are multiple vascular adaptations to spaceflight environmental factors, which in turn may affect organ physiology as well. Now, what about the GI immune state? From the same GI tail suspension study I previously described, I quantified the immune cells that trigger immune responses, called MAC2 positive cells, representative immunofluorescence figures here. In summary, as described in this figure on the right, in local GI tissue in the mesentery, there is a decrease of these triggered MAC2 positive immune cells. How does this relate to the GI immune state? Well, these MAC2 positive cells are responsible for mounting an immune response, and a reduced number or expression of these cells suggests immune impairment, perhaps linked with lymphatic dysfunction. Up until now, I've touched on low Earth orbit mission-related cardiovascular immune experiments, either from space shuttle experiments or ground-based analog experiments. A significant difference between these experiments and future lunar missions will be astronauts being exposed to deep space radiation. On their journey to the moon, they will travel beyond the Van Allen belts, illustrated here, which does not offer deep space radiation protection on the moon's surface. So what is our knowledge of GI cardiovascular and immune adaptations in the context of a lunar mission? Well, this blank slide essentially describes our knowledge in this area. With the background I provided of the GI system and its vascular and immune components, perhaps we may appreciate not only how increasing our knowledge in this area would enhance our space biology knowledge, but also, in further investigation of these areas, learn of potential medical risks astronauts experience on their journey to the moon. Indeed, of our limited knowledge of lunar mission adaptations, this study described there may be cardiovascular adaptations specific to deep space radiation exposure, and that of all the astronauts that have flown to space, the ones specifically that have traveled to the moon, the Apollo astronauts, had elevated cardiovascular disease risk compared to other astronauts. And the perspective that we gain from our travels into space is also beneficial for our studies here on Earth. For example, a majority of cancer patients receiving radiation therapy treatment experience side effects from radiation therapy. One side effect, imaged here on the left. This swelling in this patient's right arm versus her left, though which an observer may consider mild. However, there is no cure for this phenomenon. It is also significantly underdiagnosed. Because there is no cure and underdiagnosed, this swelling will often develop and become more severe, especially if not managed early, reaching a point where patients' limbs use may be very limited and lack utility entirely. Millions of individuals in the United States and across the world suffer consequences with this condition for the rest of their lives. Astronauts also experience a form of swelling that some hypothesize is partially due to space radiation exposure, perhaps for similar reasons as patients receiving radiation care. I hypothesize there to be some overlap between what is occurring with patients and with astronauts from a basic biological perspective during my doctorate, and discovered for the first time that the biology of the specific cells related to this condition in response to radiation exposure is very unique. To illustrate this, these two images on the right side of the slide are those specific cells I mentioned. In the middle here of the untreated group, and there's the other group being exposed to radiation. The ones here look as they should, while the ones on the right are how they appear after radiation exposure. You all may note the visual difference, which shows the direct effect of radiation. This basic experiment, the first of its kind, provided the first steps and clues as to what is happening to both patients and astronauts. In short, I will leave you all today with my most important slide, highlighting just a few individuals and programs that provide me the opportunity and experience I've had to share with you all today my role as part of our human space exploration efforts. Thank you for your time and attention. If there's time for questions, I'd be happy to address them.